And if it's okay for everyone, we will continue with the third presentation of this part by Dr. Saaid SAFIEDDINE. Saaid, you are also working at the PASTEUR Institute and you are also a research director. Your talk will be about the gene therapy and reversal of hearing damage, and we are almost on time. So thank you, Saaid, for joining us and for presenting today.
So thank you very much for the invitation. I'm pleased really to take a part of this very exciting short symposium. So I want to can I go back? Oh, here. So as the title actually is, it's I'm hoping that I'm going to convince you that in near future gene therapy will not only prevent but also cure some form deafness. That's what my team is trying to do actually. So this is a story of our lab. For 30 years now, we've been collecting data from patients suffering from deafness, and we've been isolating DNA, sequencing, and finding mutations involved in deafness, and generating a mouse model that help us to decipher the physiopathology of the mutation, and that's also identified cell target to really frame down the molecular mechanism of the deafness. And this our team now, I think Aziz also is trying with us to take this project one step further to restore, to correct this deafness who's involved defective genes. Of course, the aim is to transfer this therapy to patients. So those are a few genes that we are working on. The autophilin, as you know, is involved in synaptopathy with deafened benign deafness, and also deafened B16 involved stereocylin, myosin 7A, which involved responsible for SR1B, and other genes. Those are one of the most popular genes that are working on, and also, of course, connexin 26. And as you can see in these slides, and also most of the genes identified are expressed in the sensory cells, which explain why most of the genetic deafness are really severe to profound, because the target really the main actor in the auditory transmission to the brain. I think this you know about this the burden of the deafness. It affects actually it's 460 million people worldwide, and clinical prevalence in newborn is one of 700 newborn. Of these, 70% are genetic causes, and today I think Aziz can correct me, but I think there are 106 genes identified, and there are also several hundred written identification, which highlight, I think, the potential of gene therapy as an alternative or as an optional additional option to deal with deafness. Still, today there is no, and I want to be a little bit provocative, but so far there is no curative treatment. The patient has to deal with either prosthesis or cochlear implant for severe to profound deafness. But there is, I note here, there is a room for improvement for cochlear implant, because the cochlear implant contains about 12 to 24 electrodes, and in the human inner ear, there are about 30,000 neurons. So it's really very hard to imagine that with 12 to 24 electrodes, you can stimulate all the neurons that exist in our ear to compensate, to restore the normal hearing. Then I think the gene therapy for deafness, it's promising option based on the number of the gene involved in deafness, and the number is increasing every year. and also they are mostly monogenic. So it's very easy to target this gene, to target them if you have the right tool to target the defective cells in the organ. So what is gene therapy? I think there is a trivial definition. It's replace a gene, defective gene. You can replace it. If it's missing, you can alter it. If it's gain of function, some gene when they are overexpressed, they become toxic. And you can also supplement it if the gene is anomaly down-regulated. So you have to balance this deficit, the defective gene that is responsible for the disease. And you need this one tool that you need. You need the gene. You need to know the gene, of course. Here, the human genome now it's very renowned, and you know the defective gene. You design a therapeutic gene. It is identical to the defective gene, and there are also now people working on mini genes, that therapeutic gene that can restore also the function of the larger genes. And this gene, you put it in a vehicle which can vector that can have very good, I would say, efficient GPS that can target the right cell to deliver the defective gene and restore the function of the defective cells. And of course, in our case, is the hair cells. I think in our case, is the hair cells involved in deafness, or vestibular hair cells. For example, we need a vector or vehicle to deliver the I could change my laser. We need a vector to target vestibular cells, inner hair cells, or inner and outer hair cells. So that's why it's difficult to really deal with this part of the therapy, because each deafness has its own defective cells, sometimes inner hair cells, sometimes outer hair cells, sometimes both cells, sometimes vestibular cells. So it's very hard to imagine that with one vehicle you can target all of these cells. So there is a lot of effort that we are employing now to identify a virus or virus that can virtually target any cell in the organ, in the inner ear, supporting cells, inner hair cells, outer hair cells, and vestibular cells. And that's what we are working on. So this is just to show you that the gene, how human genetics help gene therapy to evolve. You see, these are the gene identified genome, actually the knowledge about the human genome during years. And this is actually the publication about gene therapy. And you can see that there is a slightly shifted compared to the human genome sequence, which we know that to understand the pathophysiology of gene, we need time. That's why we have this shift. And now that gene therapy is paper are increasing significantly every year. And as I said, we in our lab, what we are trying to do is trying to work on several mouse models for human deafness, and to treat the deafness for each mouse model, and find a way how to translate this therapy in mouse model to patient. Of course, we have now clinicians in the team that are working on this part of the project. So as you said, there is gene therapy. In theory, it's simple. You just have to replace the gene if you have the right vehicle. But the challenge to translate gene therapy from mouse model to human, they are very complicated. For example, this is the therapeutic window. As you may know, the mouse born deaf, and they start hearing about P12, around P12. However, the human, the hearing start in utero, which basically exclude this zone, as I told you, I call it here zone of prevention. If you do this gene therapy during this period, it corresponds to a preventive therapy in human, while it could be curative in mice. And the most plausible time window to be transferred to the human is from, I would say, from P20 to up to year of life of the mice. So if you carry out translational research in this green period, we will be able to translate the gene therapy to human. So the prevention. Can gene therapy prevent deafness and balance disorder? For example, in Usher Atakaiziz, he presents nicely Usher 3A, and we have, this is another mouse model, Usher 1G, which is one of the most severe of Usher syndrome, characterized by profound deafness as shown here, and also a balance defect. I think my video is not working, but this mice is turning out, I think as you saw, very nice mice also with vestibular defect. And this defect is due to, the deafness is due to the disorganization of the stereocilia of inner and outer head cells. And same thing is happening to the vestibular system, the vestibular head cells. You have here the vestibular stereocilia are very nicely organized, circus pattern, while in the H1G mouse model, the stereocilia just collapse. That's what triggered this balance defect. So can this phenotype be prevented by gene therapy? So what we did is make a therapeutic transgene. Here it's schematized here. We put just CDN coding for the protein SANS, which is encoded by H1G gene, and just stick it to GFP to follow the expression of the therapeutic gene in the cells, and we inject this transgene to the inner ear of P2 mice. Just here, I'm not sure you can see this is a patch paper that's going through the round window membrane of the P2 mice, defective for S1G gene, and we monitor. I think Aziz, he presents many ways to evaluate gene therapy outcome using vestibular behavior, capacitance, and mechanotransduction process, and we can many tools that we have now to evaluate to what extent gene therapy restore the function of the inner ear. And I said, we are in this red zone, which is zone of prevention, because it still corresponds to in utero in human, but still at the mice here, this is P2, and you can see that the vestibular head cells stereocilia are really collapsed. So when you treat these mice, the stereocilia was like this, they are really collapsed. And the question is, if you deliver this gene, some gene to these cells, it can be restored the structure of the stereocilia. And the answer is yes. You see here, this is the mice, P2 mice. When we treat it with therapeutic transgene, and this is the treated mice. You see, we went from here to here, and as you can see here, we restored completely the staircase pattern of the stereocilia vestibular head cells, and also the function as shown here in this video tracking, as it also show a very nice video, and also traces. And you see this is green traces are mouse treated with transgene therapeutic H1G transgene, and we restore this movement in the cage of the mice to near normal wild type movement. And also we collaborate with Mathieu Berenec at au Saint-Père, and he has this platform that can stimulate the different part of the vestibule, and this is canal test and this otolic test, and you can see here that in both cases we restore the function of this organ near the wild type mice, which suggests that at least in the vestibule we can restore very severe damage of the hair bundle of the vestibule. However, the hearing, as in mentioned, that this is the restoration was not to the wild type level, but there is improvement. These mice supposed to be profoundly deaf, this red line here, but they are not because of the treatment. And we have an idea why it's that way, because there are two ways. Either at P2, the cell was so much damaged that there is no way to recover all the cells. And the other thing, we know that the virus we used here in this case, it did not infect 100% of the auditory head cells, which is probably one of the cause that led to this partial restoration of hearing. But still, the deafness is not profound. So this is the conclusion of my first part. So I think I convinced you that gene therapy can restore the vestibular function even if it's severely damaged. But the hearing is partially restored, and that we know why, because probably we need a much powerful vector to target 100% of the defective cell, which is not, we don't have it yet, but we are working on it. And this is the curative gene therapy for deafness. So as I told you before, we have to work at this zone, which is called zone of translation. These are the most transposable time window to human patient, because we are working after P20, and P20 corresponds to postnatal newborn human. Of course, we need a mouse model to provide the proof of concept that the gene therapy can reverse deafness already installed in the patient. So we chose this mouse model, which is deaf and benign, involved otophilin. I think Didier talked about it this morning, which is responsible for a synaptopathy. The hair cells are here, as you can see here, inner head cells, outer head cells, rows, but the mouse are deaf. So we have the organ intact, except they are lacking otophilin, which is the calcium sensor of neurotransmitter release. So the cells are there, but they're unable to release glutamate to activate the primary auditory neuron. So this is the ABR, a trace of ABR. This is wild type mass, expressing otophilin in the RSS here. And this is lacking otophilin, and the ABR are completely flat. There is no auditory wave showing a profound deafness. And the idea is, can we reverse this phenotype by delivering otophilin transgene, therapeutic transgene? So that's what we did. We just packaged the otophilin CDNA. It's more complicated than that, but the idea is just we put the CDNA in a virus, and we have a viral preparation that we inject in this mice at P30, which is corresponding to between 6 and 8 years of a child. And we were able to restore the expression of otophilin only in the head cells, where otophilin is supposed to be expressed. Otophilin is not expressed in outer head cells, which is good for us, because that's the proof that this virus targets only the inner head cells. And this is a proof that only the inner head cells have this normal distribution of the ribbon. You see, this is green cells, expression of otophilin. If you look at the synapses, are distributed nicely at the basolateral area of the inner head cells, while this cell is not infected. The ribbon are clustered somehow at the basal part, which suggests that this cell probably does not have enough of otophilin transferred by the virus. And of course, the hearing was restored because these mice were deaf at P30, and we treated, we recover very nice ABR traces, nearly at the level of wild type, which is was encouraging for this kind of deafness, at least in mice. And also we study also the longevity, and this is more than 20 weeks, and the recovery was stable. We inject just 2 microliters of viral prep. It's not that much, and the hearing was stable for more than 20 weeks, which is very encouraging for translational research. And this is my conclusion. So this is just to state that gene therapy can not only prevent profound deafness and restore the function of the vestibular system, but also can, for some deafness, can reverse the phenotype, even if it was declared way before the treatment. So I want to thank the people who are involved in this project, Charlotte, Calvé, Alice Santos, and with the help of many others, Baptiste, Pliant, and Jérôme Nouveau also used the clinical ENT. He's working on the non-human primate part of this project, and also Sylvie Way and Laurent. And I would like also to thank people, Jacques Boutet, he's always helping us for many tasks, Nicolas Michelski, Andrea, Lily, Vincent Michel. Sorry about that, I forget his name. Sorry, Vincent, and Christine. And we are collaborating also with Mathieu Berenek and in Saint-Père, Lawrence Lastig in Columbia University, and Omar Akil in University of San Francisco. And also our funding, l'ANR, Institut Pasteur, Fondation pour Audition, and also Investir Avenir. Thank you.
Thank you very much, Saaid, for your very nice presentation and the very promising result you presented today. Is there anyone in the audience who would like to ask a question to Saaid? No. Yes, Patrick Verando.
Again, I'd like to know, Amir, if you know if the technology of CRISPR-Cas9 has been checked in this to restore the expression of defected protein.
So CRISPR-Cas9
kind of viral, vector.
Yeah, it has been done for the TMC1 deafness, but CRISPR-Cas9 it's just to down-regulated the bad allele. It's the advantage for the CRISPR. You cannot correct a gene because the cells are post-mitotic, and you cannot take advantage of the recombination to repair the missing part of the gene. But you could do also what you can do with it. You can, for example, if you have some dominant genes that became dominant and lead to progressive deafness, for instance, you can use CRISPR-Cas9 to inhibit the bad allele, the mutant allele, while keeping the healthy allele without normal. That's one way it has been done for H1C and TMC1, and it works very nicely. Yeah.
Okay.
But you cannot correct a gene. You can just down-regulate a gene that has gain of function and became toxic for the cell, for the hair cells. Yeah.
Okay.
But the tool is the same. The package, the vehicle is the same. It's the virus.
Yes.
All the
but you can act, precisely on a certain part of the genome and of the defected genes.
Yeah, because that's for TMC1. It replaced just one amino acid, well, not one nucleotide. In TMC1, just one mutation, one nucleotide, and you were able to target that allele, to delete it, and keep the allele, healthy allele, functioning, and the restore hearing. Yeah.
Okay.
And it is very promising technique for sure. Yeah. For all the gene that are for gene that gain function after mutation, those are the target for with CRISPR-Cas9 tools. Yeah.
Okay. Because there is also some problem of off-target, for all this technique of gene therapy.
Well, now the off-target, now there are very, very efficient tool to select really the guide RNA and also when you have a guide RNA, you compare the whole genome and you can to make sure that there is now sequence that are going to bind this guide RNA except your target gene. And I think this is a very, very, very good progress has been made in terms of software and synthesis, to have a very efficient, a very specific gut RNA to target the defective gene.
Okay, thank you.
Okay.
Thank you. Is there another question? Alice, yes?
I have a question about humans, because usually deafness is rarely detected very early. It's detected very late. And are there some possibility to have tests to detect very early, and in your purpose to apply those kind of therapies, you should be able to detect very soon the problems?
Well, I think there is now in Europe the screening for deafness child is very early. In the first 3 months, we have the information, but 3 months for the mice is much younger. So that's why we need to work on in a therapeutic window very specific to each deafness actually. For benign deafness, actually we are in clinical trial now. We are with collaboration with Necker, la PHP, and also Sensorion and la Fondation pour l'Audition. We are now in the process of making a therapeutic gene to treat deaf and benign patients. I hope within 3 years we'll have some positive outcome. But because we have the proof of concept that the organ, the hearing organ, is intact up to 1 year in the mice, which is nearly 10 years in human. So we can go ahead and try gene therapy in human, which is not the case for Usher syndrome because it's so dramatic. So we need more different approach. Okay, but now it's in Europe. The screening for deaf children is very systematic. Okay. You go to the hospital, you give birth. So 2 months later, you come back to check the hearing of the child. Yeah, it's systematic. Yeah.
Okay, thank you.